So good morning to all. As today we are going to discuss about DNA replications. Before going to the topics, you should know about what is the central dogma and DNA replication. The central dogma, it is nothing but the three processes. One is replication, transcription, and translation process. What is the replication? The replication is DNA itself is converted to form the new DNA. It's called as the replication. What is called? The DNA itself it will to form the new DNA. It's called as DNA replication. And what is the transcription? The DNA is converted to form the RNA. It's called as a transcription. The RNA is converted to form the protein. It's called as a translation process. Again, I tell you the DNA itself to form the new DNA is called as replication. The DNA is converted to form the RNA is called as a transcription, and RNA is converted to form the protein is called as translation process. Okay, now we know the topics should know. I already know about what is the properties of the DNA. So DNA is a double helix. It is present for the four nucleotide bases. One is adenine, guanine, thymine, and cytosine. These are the four bases are presented, and sugar and phosphate is form of the backbone of the DNA. The bases are lies between the backbone bases. What is the bases? Adenine, guanine, thymine, and cytosine. Purine and pyrimidine bases lie between the backbone of the DNA and held together by linkage between the adenine and thymine. Two hydrogen bonds is presented, and guanine and cytosine is presented for a three hydrogen bond. You can see that the picture you can see the third pictures. One is present for the left side, the adenine, and sugar and phosphates are presented. In between the line between the backbone of the DNA, the guanine and cytosine is presented, but the two bases are linked by the three hydrogen bonds. And adenine and thymine is present for the two hydrogen bond is present. So that is one of the strong bases in compared to the adenine and thymine bases. Okay. So next one is the base pairing rule. What is the base pairing rule? So adenine and thymine. Adenine is pairing with only for thymine, and guanine is only the pairing for cytosines. This one is DNA. This pairing only for DNA. The adenine is pairing is replaced by thymine, and guanine is replaced by cytosine. These are the pairing of the DNA. What about that RNA? The adenine is replaced by the uracil. Again, I tell you the adenine is replaced by uracil, so the thymine is not present to the RNA. Again, tell you the base pairing only for the adenine is replaced by the thymine, and guanine are replaced by the cytosine. So we can see that in the RNA, the adenine is replaced only for uracil. It's not presented for thymine in the RNA base pairs. Okay. So this is the structure. We can see that the thymine and cytosine is pairing with adenine, and the cytosine is pairing with guanine. The double side is present for sugar and phosphate group are present between the right and left side. So DNA standard is direction is the one end is different from the other end. So the DNA is first standard move to the 3 to 5 direction. Just in the second standard doesn't 5 to 3 direction. So this one is the opposite direction. So first standard move to the 3 to 5 direction, but the second standard move to the 5 to 3 direction. These two standard are anti-parallel and run in the opposite direction. The complementary result and important to the replication system. So the two strands are anti-parallel, they run into the opposite direction. The complementary result and important for the replications. Next one is the nucleus. You can see that it's already told what is called the DNA is converted to for the DNA itself to form the new DNA. It's called as replication. and the DNA is converted to form the RNA. This process called as a transcription, and RNA is converted to form the protein is called as a translation. Please noting that what is called as the DNA, it converted to the RNA, the process called as the transcription, and RNA is converted to form the protein is called as a translation process. So after the translation, the proteins are released. The protein after the modification and the target to the particular and specific tissues. After the post modification of the protein, they enter into the specific tissue and the target tissue of the organs. So it's clear. So the first slide can show about what is that DNA, what is called replication, and can tell us, it's already seen about that what are the bases are present to DNA and what are the sugars and phosphate presented for the DNA backbone, and how it attached with how many hydrogen bonds presented between the adenine and thymine and guanine and cytosine. So now you can see about what is the central backbone. Next, move to this next slide. The genes or information transfer. Okay, the genes or information transfer, you can see that gene A, gene B, and gene C. After the post modifications, you can carry the information to produces for protein A, gene A to carry out the transfer information and to secrete the protein A, and gene B carry out the transfer of information and secrete for the protein B, and finally the gene C carry out the protein C and secrete for the protein C. Again, carry the information and secrete the protein C. So the gene is a sequence of nucleotide within a portion of the DNA that code for a peptide or functional RNA. All genes called as, all the genes are called as genome. Gene is a sequence of nucleotide, but genome is the all genes. All the gene is called as genome. Only one gene sequence of nucleotide is called as gene. Okay, next one is Nicholson-Stone experiment. Nicholson experiment is already known and read about, I think already previously seen in the 12th class also already seen. Again, once again as here explained what is the bacteria were grow in the medium contained in the N95. Bacteria were grown in the N95. The DNA was labeled with the heavy nitrogen. Again, it is the bacteria is growth and the DNA was labeled with the heavy nitrogen. The first generation, what can is that? The first generation N14, and all the DNA molecules were off-labeled here. And the second generation, the generation off-labeled, finally unlabeled DNA in molecular were presented. We can see that structure. The first one is the N95 molecular heavy DNA is presented in the first generation. What can I do? The off N95 and N14 intermediate DNA density DNA is formed, and the second generation 50% of N95 and N14 intermediate density formed, and 50% of N14 sorry, N14 and N14 DNA is formed, and finally the N14 is secreted the 2 DNA and intermediate form the 50% of N95 and N14 and the 50% of secreted for N14 and N14, and finally you will get it the unknown labeled DNA. Is clear? So first what can you do? N15 and N15 nitrogen, the heavy DNA is present in the first generation started, and the second this one is a starter material after growth of bacteria, and the first generation what will do, the N15 and the N14 are intermediate density to form, and after the 50% of final with the N14 and N14 the label of DNA is formed, and 50% of intermediate density of DNA is formed. What can I do? And that allows Generation third, what can I do? The N14 is 100% of N14 DNA is formed, and N intermediate DNA tends to form 50% of N14 and 50% of intermediate DNA will form. After the final generation, what we will do is all the DNA form unlabeled DNA molecules are were present. So these are the experiment is called as Michelson-Stoll experiment. Okay. The base pair rule already told the one side DNA standard is presented A, sorry, the A is replaced by T and D are replaced by adenine. That means thymine is replaced by adenine and adenine is replaced by thymine. So again, the cytosine is replaced by guanine and guanine is replaced by cytosine. This is the base pair ruling. Suppose any base pairs are mutated here, any bases one is this DNA is pairing with only the adenine and adenine is only the pairing with thymine and cytosine is only pairing with guanine and guanine is only pairing with cytosines. But any mismatches base pair, for example, the guanine is replaced by the cytosine or cytosines are replaced by the guanine. This process is called a single nucleotide polymeration. So only one base pair or only one base pairs are replaced by other bond. Suppose the three nucleotide or deleted or against or inserted, this is called as mutation. Okay. And the next one is the semi-conservative model is already told the same like you can parent DNA standard. This one is the parent DNA standard. What can you can separate it the one parent gene separated creating the two single standard and each standard is used as a template for a complementary standard shown in the you can see that this one is the parent DNA is separated for 50% of parent DNA and 50% of new DNA standard are formed. The two standard first the 100% of parent DNA is separated to the single standard and the single standard combined with the new DNA standard. What can I do? The 50% of new standard parent DNA will form 50% of new standard and 50% of intermediate DNA form. Again to separate the other bond also 50% of form intermediate and 50% of new standard are formed. It's clear. So once more I can tell you what is the parent DNA. It is separated the single DNA to single DNA. That single DNA are separated the new DNA strand are formed. This intermediate to DNA standard it form 50% of newly synthesized DNA form unlabeled DNA standard form and 50% of intermediate DNA will form and here also another standard also the 50% of new DNA standard form and 50% of intermediate form and finally what can I do? Finally what will do and what will occur it is 100% of unlabeled DNA molecule were presented unlabeled. So the first generation is 50% form and the second generation what will do the 50% is converted totally unlabeled DNA standard were formed in the second generation of DNA. This is called as a semi-constitutive. The parent DNA is contained in two single DNA. The two single DNA is after separating for the parent DNA, it will be separated the two single DNA. The two single DNA is attached with the new DNA that each intermediate DNA it will form 150% new DNA and 50% of intermediate DNA. And finally, to form the newly presented and unlabeled molecule of DNA were presented. Next one is the basic requirement for the replication. What are the substrate are needed for the replication? One is DATP and DGTP, DCTP and DTTP. It means adenine triphosphate, deoxyguanine triphosphate and deoxycytosine triphosphate and deoxy thymine phosphate. So these are the four substrates are needed for the basic requirement for the replications. So another product is template. Is already told that the template is the parent DNA. You can see that each standard of the DNA molecule serve as a template standard for the synthesis of a new doctor standard. So each standard of the DNA molecule serve as a template standard for the synthesis of new doctor's standard. The template is required to direct the addition of the appropriate complementary nucleotide to the newly synthesized DNA standard. So the parent DNA is contained two single DNA that is separated for the two single DNA. It will form newly synthesized doctor standard. So the template is required to direct the addition of the appropriate complementary nucleotide to the newly synthesized DNA standard. Next one, what are the enzymes? The DNA polymerase and topoisomerase. The two enzymes are involved. One is DNA polymerase, another one is topoisomerase. This is the most important enzyme in this DNA replications. The enzyme, one is topoisomerase. There are two type of topoisomerase are in. One is two type of topoisomerase. One is type 1 and another one is type 2. What is the difference between the type 1 and type 2? The topoisomerase type 1 they catalyze the relaxation of the supercoiled DNA by breaking just one strand of DNA, whereas the type 2 cleaves both standard. You can see that the topoisomerase type 1 they catalyze the relaxation of the supercoiled DNA by breaking the just one strand of the DNA, whereas the type 2 cleave the both standard. The topoisomerase 2 also called as the DNA gyrase. It looks like it's topoisomerase, otherwise it's called as gyrase enzyme. Can introduce the negative supercoil to the DNA using the free energy from the ATP hydrolysis. The primers, the next one is the primer. Now you can see that what are the substrate and what are the product that are needed for the DNA replication. First one is the enzymes, next one is that template. Now move on to the primer. What is the primer? It is nothing but this contains 10 nucleotide in the length, the small length of the DNA. This is the primer which is required for the DNA synthesis. It's a short it's a short piece of the RNA. It is contained the 10 nucleotide in length. It is formed by the DNA dependent RNA polymerase is known as primase enzyme. It is formed by the DNA dependent RNA polymerase is known as primase. The primer is synthesized from the 5 direction to the 3 direction using the DNA as a template. The primer where it is synthesized 5 to 3 direction using the DNA as a template. The primer is nothing but it's kind of short piece of the RNA. That RNA is containing 10 nucleotide in the length. It is formed the DNA dependent RNA polymerase is known as primase enzyme. The primer is synthesized to 5 to 3 direction of the 3 direction using the DNA is act as a template. The DNA polymerase initially add a deoxyribonucleotide to the 3 direction hydroxy group of the primer and then continue to add the deoxyribonucleotide to the 3 direction end of the growing strand. So what can the DNA polymerases initially add to the deoxyribonucleotide to the 5 direction of hydroxyl group of the primer and then continue to the deoxyribonucleotide to the 3 direction end of the growing strand. So what can you do? 5 to the DNA polymerase is synthesized to the 5 to 3 direction of the growing strand. So now we can see the DNA polymerase enzyme. The DNA is copied by the replication machine that is travel along the each replication fork. You can see that what is the replication fork after the slide. You just note on that what is the DNA is copied by the replication machine that travel along the each replication fork. One of the most important membrane of the complex, the DNA polymerase. The DNA polymerase enzyme is one of the main important enzyme in the DNA replications process. So what are the types are presented in the eukaryotic and prokaryotic? The prokaryotic contains 3 types, 1 and 2 and 3. It's the prokaryotic and the eukaryotic. What is the alpha, epsilon, beta, gamma and delta. Okay, so prokaryotic 1, 2, and 3, this alpha, beta, gamma, epsilon, and delta is present for the eukaryotic. What is the function for this group? The primase activities, the gap filling, then synthesis of lagging stranded. What is the function of that? Program DNA polymerase 1 and alpha, the primase activity, the gap filling of synthesis and logging standard. So in between that primase activity based by the gap filling and synthesis for the logging standard. And what is the polymerase DNA polymerase synthesize polymerase 2 and epsilon. It acts as a proofreading and repair of the DNA replications. And beta, what can I do? The DNA repair, it acts as a DNA repair. And gamma, it can act on the mitochondria. It only acts as the mitochondrial DNA synthesis. Polymerase 3 and delta is highly possessive and leading the standard of synthesis. So clearly that what is the prokaryotic type? DNA polymerase 1 and DNA polymerase 2 and DNA polymerase 3. And the eukaryotic alpha, epsilon, beta, gamma, and delta. So what are the functions for that prokaryotic for DNA polymerase 1 and eukaryotic polymerase DNA polymerase alpha? It acts as a primase activity, gap filling, and synthesis for the logging standard. And the polymerase 2 and the epsilon, it acts as a proofreading and the repair of DNA repair mechanism, and beta is only act for the DNA repair mechanism, and delta, gamma, it acts as the mitochondrial DNA process, and delta is highly processive and leading to new DNA standardized synthesis. DNA ligases, it acts as the RNA primase must be removed and replaced by the DNA. You can see the sealed by the ligases after the removal of DNA segment and filling the gap with the DNA. There remains point in the DNA, the backbone, where a phosphodiester is broken. You can see this picture. You can see that the phosphodiester matter are removed. If they removed, this spot are removed and sealed again to the missing the phosphodiester bond. It is helpers to the DNA ligase. What is that? We have to fill in. We have to that replace. These are filling in the gap between the first standard to the second standard. What can it after the removal of the DNA segment, the filling in the gap with the DNA, there remain the point of the DNA backbone where the phosphodiester bond is broken. So what can it? The phosphodiester bond is broken. The missing the phosphodiester bond at the replaced by and attached with again back by the DNA ligase enzyme. We can see the missing phosphodiester is shown in the picture. The missing phosphodiester bond. We can show here mismatch for the missing to the phosphodiester bond, and after the DNA ligases act, you can fill the gap between the two bond. So this is the sealed by the DNA ligase. This specific particular position are sealed by DNA ligase enzyme. It's clear what is the function of the DNA ligase? The remove must be that the missing to the phosphodiester between the two phosphodiester bond between the backbone of the phosphodiester bond. You can missing the bond are synthesized by the DNA ligase and sealed by the DNA ligase in the missing phosphodiester bond. This is the DNA ligases process. Now the next one is the step involved in the DNA replications. One is identification of the origin of replication and unwinding of the DNA to provide as a single standard DNA template and formation of the replication fork. What are the steps? One is identification of the origin of the replication, unwinding of the double-stranded DNA to provide as a single-stranded DNA template, and the formation of the replication fork. And fourth, the initiation of the DNA synthesis and elongation, formation of the replications buffer with ligation of the newly synthesized RNA segment. And sixth one is the reconstitution of the chromatin structure. So what are the steps for the replication? First one is identification of the origin of the replication, and the second one is unwinding of double-stranded DNA to provide as a single-stranded DNA template. And the formation of the third one is the formation of the replication fork. And fourth one is the initiation of the DNA synthesis and elongation process. And the fifth one is the formation of the replication buffers with ligation of a newly synthesized DNA segment. And the sixth one is the reconstitution of the chromatin structure. So please note in that these are most the six steps are involved in the DNA replications. So the identification of the first, you can see the identification of the origin, the seg. Where is the start of the replication process? First one, you can start it from the yellow color. These are the origin. The DNA replication started with the recognition of the site origin of the site of the origin of replication. The main origin, the autonomous replicative sequencing have been identified in the mammals. The first one is where it started. You can see that the yellow color can point out this here DNA standard. So this process and specific and a particular position or standard to started from the DNA. Then what can you do? The replication started the point of the origin of the replication is started to form the bubble like. So this is called as replication bubble. Look like the bubble in the DNA standard. It is the bubble structure is called as replication bubbles. And finally, I have to separate it for the double stranded. You can see that the parent DNA one is formed, and the one is the single new doctor DNA is formed. So 50% of parent DNA and 50% single new DNA strands are formed. So first you have to identification where it started for the DNA replication, and the second step it will to form the replication bubbles. The replication bubbles is nothing but after the origin of the replication, the origin of the replication started look like a bubble structure. It's called as a replication bubble. And after that, what is the to separate it for the two single DNA, parent DNA, and attached with the two. And 50% of single, 50% of parent single nucleotide, single DNA standard, and 50% of newly synthesized DNA, doctor DNA standard it will be formed. The replication. So you can see the replication bubbles. What's the replication bubbles? The two complementary stranded of the DNA separated at the site of the replication to form the bubbles. Again, it is the two complementary DNA stranded. The DNA is separated at the site of the replication to form. This is the site of the replication. Two site of the replication. Single subdivision is formed. It looked like a bubble. Okay, this replication to form a bubble. The multiple replication bubbles are formed in the eukaryotic DNA molecule, which is essential for the replication process. So this is the DNA polymerase enzyme. This is starting of the DNA publication process, bubbles process. This is the DNA polymerase. We have to end of the DNA polymerase enzymes that act as here, and parent DNA is black and red color is doctor DNA. After that, replication bubbles will occur to separate it for the two single DNA of parent DNA and two single DNA of doctor DNA. The 50% of parent DNA is formed and 50% of single new DNA doctor DNA strands are formed. But here the multiple replication bubbles were formed in a eukaryotic DNA process molecule. I can tell you the multiple replication bubble is formed in the eukaryotic DNA molecule. Next one is the replication fork. So what is the first one is replication bubble, another one is the replication fork. The separation of the two stranded of the parent DNA in the formation of the replication fork, and another one is the replication fork is along with the parent DNA as the doctor DNA molecule are synthesized. That is separated for the two DNA stranded of parent DNA is on formation of the replication fork. Another is the replication fork moves along the parent DNA as a doctor DNA, and the DNA molecules are synthesized. You can see the one is the parent DNA, another one is the fork regression. You can start it from here. One is Another Dr. DNA synthesized from the 5 to 3 direction. Here are the opposite for the 5 to 3 direction into that. First one can the replication for parent double helixes. The leading standard, another one is logging standard. Leading standard is continuous synthesis for the Dr. DNA, and the logging standard is discontinued to form the logging standard. One is the DNA primer and Volkswagen fragment and logging standards. So the replication fork moves along the parent DNA as a Dr. DNA molecule are synthesized. You can see that next one is discontinue to form the leading standard and continues for the leading standard, and logging standard what can be discontinue to form the logging standard. The discontinue the logging standard is called as Ogasagi fragment. Again tell you there are two standards form. One is leading standard, another one is the logging standard. The leading standard is continuously synthesized and the logging standard is continuously synthesized. This continuous synthesis process is called as Ogasagi fragment. The replication bubble to form this one is act on the eukaryotic. So many replication bubbles are formed. DNA polymerases alpha, beta, delta, epsilon, and gamma, and logging